Welcome to Freeslate's webinar on Automated Solutions to Accelerate Drug Development. This is the 8th webinar in our ongoing series on this topic. My name is Dave Imani, and I'm the Product Marketing Director for Life Sciences at Freeslate, and I'll be your host today. Today you'll hear a presentation on some formulation work we performed using Freeslate's Biologic Formulation System. We're pleased to have Dr. Russell Burge from Freeslate join us today. Time permitting, we will conclude today's webinar with a brief question and answer session at the end. Before we begin, let me invite you to submit questions using the Q&A menu on the right-hand side of your audience console. In the Q&A window, you can type in your question and press the Submit button. Any questions that we did not get to will be answered privately in a follow-up email from Russ. Our speaker today is Dr. Russ Burge. Russ earned a Bachelor of Science degree in biochemistry from the Arizona State University in Tempe. Russell obtained a PhD in molecular biology and biochemistry from the Scripps Research Institute in La Jolla, California. After graduation, Russ received postdoctoral training at the University of Colorado in Boulder. During postdoctoral training, Russ worked on the biophysical characterization of RNA and DNA aptamers relevant to the biopharmaceutical industry. Russ then joined KBI Biopharma as a scientist, where he worked on more than a dozen biopharmaceutical development projects. Currently, Russ is an application scientist at Freeslate. The title of Russ's presentation today is Highly Automated Processes for the Assessment of Protein Formulations.
Okay, thank you, Dave, for the kind introduction. And so I'll be talking today about a highly automated procedure that we've used with one of our automated systems to assess protein formulations. And so what I would like to do now is give you an overview of what I'll be talking about. So this webinar is meant to present a case study performed on a stable model protein, specifically BSA, which will show the utility of our Freeslate systems for both pre-formulation and formulation development of biopharmaceuticals. So this study is a hybrid study in two ways. It basically includes many elements of buffer and pH screening, as well as DOE and forced degradation and short-term accelerated stability studies. But it's also a hybrid study in the sense that it's both automated and manual. And so a big part of what the webinar today is to demonstrate the value of the automated systems in these development laboratories. Some of the general principles I'll be discussing will be how to use the automation to create formulations using liquid compounding, also to automate previously laborious and manual methods that can also be very strenuous on the analyst. I'll talk about the study planning, experimental design, and the different procedures that we use to implement the automation to work on such studies. And the idea here is to provide some insight to the audience on how the automation can improve your efficiency in the collections, storage, analysis, and your presentation of your data. And so, really, the main point here is to show how our automation systems can improve your current activities in the laboratory. Okay, so Freeslate is a spin-out of a former company called Symix Technologies, and so Freeslate owns the IP that covers the high-throughput microscale technology as well as the automation tools. We have a broad and global customer base. Many of the attendees are actually customers of ours, and you can see on the right the logos of many of our companies. We have about 85 employees, 60 of which are scientists, engineers, and developers. We are headquartered in Sunnyvale, California, and we have a field office in Geneva, Switzerland for our European customers. Okay, so as part of our business model and how we work with our customers is we have this sort of three-part cycle here, which starts with our proprietary automation platforms. So currently we're working with configurable core modules and workstations, and for this specific study that I'm talking about, I actually use what we would call a core module system. We also create custom systems for some of our customers as well. And then in the next part of this circle, we have the LEA software, the lab execution and analysis software, which basically drives the whole ability for us to perform automation. It allows us to do our experimental design. It allows us to run the automation systems as well as third-party analytics. And it allows us to analyze and report the data rapidly. And then in addition to this, we also use our scientific expertise and collaboration and innovation to work with customers and potential customers to create systems and also to work with them with the current systems they have to get the most out of the system. Okay, so basically our software split up into three. Our software suite has basically three sub-software programs. And you can see on the left here, no matter whether you use automation or manual methods, you're always going to start with this sort of general design, execute, analyze, and report type logic. And what we do is we basically have the three software programs used to perform different functions. So for instance, our Library Studio software is an array-based software program that allows for experimental design that will then be pulled into the Automation Studio. And using Automation Studio, you can control the automated systems and instruments, and it also then takes that data and moves it to the database for all devices that I'll be talking about today. So basically, Automation Studio and Library Studio can be used to run all of the systems. So really, you really only need to know two different software programs. You don't need to know the third-party software programs. And then we have a program called PolyView, which allows us to create reports from the database. And as you can see here, there's sort of three very small images of example reports. So you can, whether they are size distribution plots or chromatograms or just normal metrics or even images, you can see them in a rapid way, and you can customize your reports. Okay, so specifically the study that I'm talking about today has implications for biological formulation development. And formulation development is a critical and challenging endeavor for the biopharmaceutical industry. Now, the ideal biologics formulation must meet several requirements, including regulatory requirements. It needs to ensure clinical performance. It needs to enhance drug manufacturability. It also needs to be compatible with the chosen mode of delivery and closure system. And so specifically here I've italicized in blue maintains long-term stability. So that's another big part. It obviously plays into manufacturability. But in this case, this is one of the areas that this study is working around. And as far as the long-term stability, we know that there are different factors that can affect that of a protein formulation. So specifically pH and buffer. And then there's also sort of stressing conditions such as temperature and agitation and the like. And naturally there's other issues, exposure to surfaces and light exposure and things like that. And then here I've just highlighted that normally protein formulations have a multitude of different degradation pathways. So this is the way that we basically are able to discern which formulations are performing better than others. So now, given that information, I'll talk a little bit about the automated systems that I use to perform this sort of hybrid study. And so the first here is what we're calling the biologics formulation system. So this is just to give you a high-level overview of the system. So it's basically designed to be an end-to-end solution for the assessment of biological formulations. It has two robotic systems separated by a carousel, and then it's all enclosed in a HEPA enclosure. And so this is basically intended to be used in development labs that are performing pre-formulation and formulation development of biopharmaceuticals, both platform and non-platform. And we have designed it so that it's well suited for formulation screening and DOE studies as well as stability studies. And it also has the capability, in addition to creating the formulations using liquid compounding, it also can create sample stressing conditions, and it can be used to create the analytical samples, and it can also test those analytical samples. So the idea here is to prepare, it can actually prepare more than 200 formulations per day using liquid handling and basically using a liquid compounding method. It can screen hundreds of formulations a day depending on what types of methods you're using and what are the different parameters of your methods. And so, as mentioned before, it's basically intended to look at the different variables of the formulations, including buffer, pH, excipients, API concentration, and it's capable of doing all of that in sort of one large unit. And naturally, this is can be used to improve efficiency, identify perhaps better performing formulations. It reduces the material consumption, and I'll talk a little bit more about that later. As I mentioned, it has built-in sample stressing capabilities, and we also have third-party stressing capabilities. And another really important thing is that it has very flexible container and consumable usage. Whatever containers you're currently using, the system can basically work with them, and whatever consumables you like to work with, basically as long as it works physically with the system, you can use them. So to give you some more specifics about what this system looks like, and to recap or to be clear, this is the actual system that was used to perform the study I'm talking about. It basically, as I mentioned, it has two robots separated by a carousel. So the first robot, we're going to call that the core module number 1. So that's where the formulation preparation, sample and analytical prep is performed. It also has a Freeslate analytics that are integrated. So that's the pH, automated pH probe, automated viscosity, and automated visualizations inspection station. And then we also have a carousel that can be used to provide additional storage capacity and move things from one robot to the other. And then on the left side, we have the core module 2, which has the integrated analytical capabilities. And the next slide, I'll show you a little bit more specifics about that. But basically, there are many different methods that can be performed by the different analytical systems that are on this deck. And in addition to that, we have shaking incubators, vortexers, and heaters. And we also can use our systems to run and pull data from UPLCs, HPLCs, CSCS, and CIEF. And so we're going to move on now to some more specifics of what these robots look like. So as I mentioned before, that core module 1, this is what the deck looks like. So this is sort of zooming in on the robot. And starting from the top left here, I'll start with the 4-tip pH probe. So that's part of the arm of the robot. And then we're moving toward clockwise to the viscosity station. So that's our viscometer with sterile pipette tips. Next is the vortexing station, which can be used for both sample preparation, analytical sample preparation, and stressing of samples. The next is the cooled storage bay, which allows for temperature control below ambient temperatures. This can be used for, again, preparation of samples, but also can be used as a condition for your samples as part of your studies. We also have plenty of pipette tip storage that allows for daughtering of plates or aliquoting of material. Then we have a 6-tip pipetting system. This is an air displacement pipetter that allows for preparation of both formulations and of analytical samples. Then we have the flexible tool changer, which allows for either positive displacement or air displacement pipettes. And then we have a very large waste bin here for the pipette tips. We have a wash station, which is used to wash and clean the 4-tip pH probe. We have a heated storage bay, which can be used for both sample prep and stressing. And then we just have more plate and sample rack storage for allowing for where your vials are going to be, whether they're analytical or formulation vials. And then we have our automated visualization inspection station. And then on the left side of the robot, which I referred to as the core module 2, we have sort of this optional configurable set of different robots and third-party systems that can be placed here. So specifically, we have, starting on the left, we have incubator shakers that can be used for 96-well plates. Then we have our robotic system there with the vial plate gripper, which can basically move 96 well plates and other sample racks from place to place, and also back to the core module 1 via the carousel. And then next we have an Ixion centrifuge, which can be used to centrifuge your samples down prior to analysis. We have an integrated Wyatt Dynaprobe, DLS, and then we have molecular devices Spectromax, and finally we have a thermoplate sealer for 96 well plates. So as far as we have two different capabilities of liquid dispensing for formulation and also for sample preparation. We have positive displacement pipette tips, and we have multi-channel liquid dispensing. So with the PDT, you can basically use Eppendorf, Gilson, Rainin tips, Tecan tips, whatever you'd like there. We have the dispense ranges from 10 microliters to 10 milliliters, and this allows us to dispense highly viscous liquids. And the disposable tips will hopefully reduce contamination concerns, and certainly they can also be reused if you're using them across one type of buffer situation. And we also have repeat pipetting functionality, which allows for efficient transfers. With the multi-channel, so this can dispense 1, 2, 4, 6 liquids in parallel, has nice dispense ranges from 5 microliters to about 1 mil, and it has variable pitch that can be used to change the format if you're going from a 2 milliliter serum vial, for instance, into a 96 well plate. The tips will automatically change for that transfer. And then we have our Freeslate automated analytics. Now these are, to be clear, these operate unattended. And we have our solid state pH probe. It's a 4-tip probe. It has high reproducibility, it's robust, and it has high throughput. It can do about 2 measurements per minute. And we also have our on-deck viscometer. This it's completely automated washing and analytical steps. We use sterile tips, and that will limit the carryover between the samples. It's relatively rapid. You can do it's about 0.5 measurements per minute. It's temperature controlled for highly reproducible samples, and it has a very low sample consumption of about 10 microliters per replicate. And then we have our visual inspection station. So here it measures color, turbidity, and it can also count visible particles above 80 microns. 80 micrometers. And then it has it can do all 3 analytical tests with about 0.5 vials per minute, and it basically provides objective measurements of color and visible particles. It's a non-destructive analysis, and it also can create videos and pictures of all of your vials for review or notebooking. So now moving on to, so that's the system that was used in the different Freeslate analytics that were used. And I'm going to move on to the experimental design and procedure of the study. So in this case, it was I realize these are relatively small tables, but the point to show here is that basically we've used several different buffers, 5 buffers, and different pHs ranging from 4.5 to 7, 4 excipients with and without preservative, and we've used the BSA at concentration of 10 mg per mil to emulate some of the activities that are performed in the formulation lab environment. And so here it's basically, so this is just split up into two tables of 48. And so this is the design to show where we're starting from and how we then will create these formulations using stocks and the automation. So to give you an overview of how the procedure works, basically we can split it up here. On the left side we have automation, and on the right side we have manual operation. So this is a highly automated procedure, but it is a hybrid manual and automated procedure. So basically what we've done here is we've started by formulating the samples in the desired containers from stock solutions. In this case we wanted to use 2 milliliter serum vials, and we created the stocks and put the stocks on the deck. We used 2 milliliter serum vials, and we created the stocks on the deck. We used 2 milliliter serum vials, and we created the stocks on the deck. We used 2 milliliter serum vials, and we created the stocks on the deck. We used 2 milliliter serum vials, and we created the stocks on the deck. We used 2 milliliter serum vials, and we created the stocks on the deck. We used 2 milliliter serum vials, and we created the stocks on the deck. We used 2 milliliter serum vials, and we created the stocks on the deck. We used 2 milliliter serum vials, and we created the stocks on the deck. We used 2 milliliter serum vials, and we created the stocks on the deck. We used 2 milliliter serum vials, and we created the stocks on the deck. We used 2 milliliter serum vials, and we created the stocks on the deck. We used 2 milliliter serum vials, and we created the stocks on the deck. We used 2 milliliter serum vials, So as far as automation goes, we'll use the liquid handling and the cooled and vortexing modules to formulate. And as a manual part of the process, we need to set up our vials, our stocks, and our consumables. But once those are set up, the automation will create the formulations. And then in the end, you can take those formulations. In this particular case, they were manually capped or stoppered. And then we were then able to perform some analytical tests. So here, for instance, the on-deck analytical tests were visual inspection, so that's color, turbidity, and particle count. And then we were able to use the vial plate gripper, then we were able to use the liquid handling to take the manually uncapped vials and create dotted out plates. So aliquot material from the initial formulations into sample vials that can be used for analysis. And so I'll talk a little bit more about the different analytical methods that we used a little bit later. And then, of course, we can perform the on-deck and off-deck analytical tests. Again, you can see on the right where the manual operation is actually used to tune the system to make sure it has the materials it needs to continue forward. And then we can also use on-deck heating elements and cooling elements to stress the samples. And then in this case, we used manual operation to move the plates from instruments and modules, between instruments and modules. And then a key note here is that all of the automated operations are unattended. So once you set up your liquid handling or your instrumentation, or you want to perform your analyses, or you want to perform your stressing, you just set it up and you go. And it keeps track of what has been done to each plate. So speaking of plates, so we refer to these in our software as substrates, but in the containers that are inside the substrates. But basically, they're vial holders, sample racks, 96 well plates, as we're all familiar with. And basically, in our software, they kind of look like a bird's eye view of what's in reality. So on the top here, we see a 4x6 example of a 2 milliliter serum vial rack. And then next to that, we have 8x12. This is a 96 well 1 milliliter vial plate that's used for aliquoting material for analysis, for instance. So in this case, you can see it's protein viscosity and pH can be measured in such a plate. And then we also have 96 well plates that can be used for UV and DLS. So this is sort of how it looks in reality versus how it looks in silico. And then so to start from where the study begins, as I mentioned before, we use the liquid handling to create our formulations from stocks. So basically we're doing a liquid compounding step here. And what you can do is in the Library Studio, you can basically take that table which I showed earlier that had the 48 formulations, and you can say, these are the target concentrations that I'd like to have. These are the stock concentrations I have. This is the final volume. Calculate and create these formulations. And that's exactly what it does. So you basically just tell it what type of container, what your stock concentration is, what your target concentration is, what your target volume is, and it does everything else for you. Now, naturally, you have to tell the system which pipette tips you'd like to use and how you want to treat the liquids, but other than that, it's all automated. And it allows then for flexibility on how you want to prepare your samples. So those formulations then can be aliquoted out. We also refer to that as dottering out. You can basically aliquot material from your formulation to a target plate or vial using air displacement or positive displacement. And as I mentioned before, there's flexibility. There's no need to have to do this in 96 well formats or 4x6 formats. This is completely flexible. And then you can even further dilute those materials after testing to other plates. So for instance, the very first plate here that's analytical, this 8x12, which says viscosity and pH, basically you can go through and use the viscometer using sterile tips and it will automatically go through every single one of these samples that are highlighted in green, and it will measure the viscosity, and then it will immediately after that's done go through and measure the pHs of those targets. And then it can take that material, the very same material from there, if there's enough material, and it will aliquot out to one or more plates. In this case, you can do one of two things. You can aliquot to 2 plates, one being for UV and DLS and one being for HPLC, or you can first make a plate that's doing HPLC, and then it's going to be used for UV, or you can create plates serially. So this is all flexible, and it just depends on how important your sample is to you versus how much you want to run your samples in parallel. So for this particular study, once we had the formulations, we wanted to have some ways of stressing the samples that are similar to what formulation labs are doing today. So first we used agitation. This was performed on deck. It took about 2 1/3 day at about 750 RPMs to sort of stress the samples. This was done at nominal room temperature. We also put the samples, so then we made another set of formulations that were put through freeze-thaw. This was performed off deck. There were 12 cycles, so this is actually pretty extreme freeze-thaw stressing from -80 C to room temperature and back. And there was also heat stressing, in which the formulations, another set of formulations, another set of the 48 formulations to be clear, was heated for 3 weeks at about, or for about 3 weeks at 37, and then it was stepped up to 45 to ensure that we had some stressing occurring so we could see some differences in the formulations. And what I'd like to mention here too is that for all of these stresses, the formulated material in the vials could be kept in their aluminum racks and could be simply moved from the deck elements or even left on the deck and performed in situ. So this made it so that no extra labeling was required. When I say labeling, I'm talking about the labeling of the vials, and so that actually helps with reproducibility and sample integrity. Okay, so as far as our automated analyses go, basically here I'm sort of splitting it up between our on-deck, which we call our physically integrated analytics, and then our off-deck, which are what we call our virtually integrated analytics. And so here what we did is, as I mentioned before, once the formulated materials are capped, we use the visualization station to perform the color, turbidity, and particle analysis. And then we open up those vials and aliquot out, and then we can perform our viscometry and our pH serially, but in a fully automated fashion. And then, so that basically has to be performed 1, 2, 3. And then as far as your third-party analytics, once you aliquot the material out that you need for analysis, those can all be performed in parallel. It depends on how many plates you want to use and how many different instruments you have, but basically you can create 3 or 4 plates and run all 4 of those plates at the same time. So these are just giving you some idea of the flexibility and the architecture of how to plan some of these studies. So once the data is collected, we have an Oracle-based database that allows you to look at your data. So in this case, I'm showing data from one of those vial racks, both DLS and reverse phase data. So you can see it here both graphically and with the chromatograms, or you can also look at numerics that you're specifically interested in that you want to pull out for reports. And the way you can do that is we have the PolyView software, which basically what I'm showing here is on the left, it's just the different vials, and for one particular library or vial rack, and then I go through and show the color result, the turbidity result, and the particle result, and you can basically export this to Excel, and that data then can be used to generate reports. And these reports are completely customizable. You can pull out whatever information you want. Okay, so now I'm going to talk a little bit about the data summaries for this particular study. And so this particular table is very large and has a lot of information on it, and that's the intention. So it will be kind of small and hard to read maybe, but the point that we're making here is that there are 48 different formulations, and at T0, so our initial time point, we are performing 6 automated methods along with a few of the manual methods. And this only takes about 6 to 9 minutes per formulation for the automation, and that includes the aliquoting for the analytical samples and also the dilutions if necessary. So this, and most of that time is totally unattended. So as you can see, basically, if you look carefully through here, you can see that the pHs are more or less where we targeted them to be. Because we were using liquid compounding, some of the pHs varied a little bit more than normal manufacturing specifications. We also had manual and automated turbidity to look at the samples as they came out of being formulated. I performed manual particulate analysis and compared that to automated particulate analysis. And then there was automated viscometry was performed, and there was also color that was performed, and there was also automated A280s. So from this, we can basically see that the automated and manual methods more or less corroborate each other, and in addition to that, we were able to see, as I mentioned, the pH was basically on target. The protein concentration was a little bit lower than the target. However, this is just a calibrational error and can be very easily remedied. It also has to do with compounding of the 2 errors of error from formulation and then analytical error as well, and aliquoting error. So basically, this is just to show you how rapidly these 48 formulations can be evaluated from their 2 milliliter serum vials out to the daughter plates, and then you basically can get your information from this in about a day. So in addition to running the Freeslate analytics and the manual analytics that I just talked about, we also performed some HPLC methods to compare the T0, the freeze-thaw, the agitation, and the heat samples that I mentioned earlier. So in this case, in red is the initial time point, in green is freeze-thaw, in purple is the agitation, and in the cyan color is the heat-treated samples. And what we can see at T0, we have a relatively high percentage of main peak area, so we'll just call this purity. And so this purity is roughly 98% or so. After freeze-thaw, there are some cases where there's a very small dip, but in most cases it's within experimental error, just a few percent of T0. That's also the same with agitation and heat. I've split up the reverse phase result into a second chart just so it's a little bit easier to read. And here again we see that they're all, the percent areas are basically hovering around 98%. There are a few cases with the heat and even the T0 where it's a little bit lower in percentage, but the main part that I want to make clear here is that we see that there's some reproducibility and consistency between the different especially at T0, for instance, where we'd expect the performances to be pretty similar. We see that there are only very small variations between the samples and formulations by HPLC, by reverse phase HPLC. Now, the SEC results are kind of interesting because at T0, we can see that there's about 11 to 12% of the protein that we can see is polymer. So basically on the left side, this main peak area, that's basically monomer percent. So we're looking at about 88, 89%, somewhere in there of monomer at T0. And this is highly reproducible, as it would seem, and consistent across all T0s, as we'd expect. In the freeze-thaw, we do see that the data is on par with what we saw with the T0 and perhaps in some cases a little bit less. What's interesting is that both for the agitation and the heat stress samples, there's a slight increase in the percent monomer. Now, something might be happening to the polymer and not necessarily happening to the monomer, or this could also just be slight variations in the mobile phase, for instance, which is allowing a small increase of the monomer to be present. So in this case, it's only a few percent. But in any case, they're highly consistent throughout, and what this data is showing us is that BSA is actually, not surprisingly, a very, very stable protein. And I'll move on to SEC chart number 2, which continues this sort of trend of T0s actually has some of the lower amounts of monomer, and there's just a very slight increase with the stresses, which is normally not what you would see with a biopharmaceutical, and I would say that most of these changes are within experimental error. So here we can see that sort of consistency between the formulations and a consistency between the different stressing conditions. Now we have DLS, which was used to look at the different formulations as far as particle formulation, or sorry, particle formation in each of them. So here we're looking at percent polydispersity. So basically this can be thought of as the lower this number, the more of the light scattering is being produced by the monomer protein. So the higher the polydispersity, the more particles there are in the system. And what we see here at T0, we sort of have this somewhere between 16 and 23% polydispersity. And much like what we saw with our SEC results, the agitation and the heat, there's actually a slight decrease in the particles that are in the different formulations. So this is a bit surprising, but it is consistent with our SEC, and it's also suggesting that these formulations are working quite well with this stable protein. Now this continues on chart 2, but here we can actually see that for the sodium acetate, for instance, there were some particles being formed specifically at the 4.5 and 5.5 pHs, but otherwise we see very similar results to chart 1, which is basically between about 16 and 23% polydispersity, with the agitation and the heat stressing actually having sort of consistently lower results compared to T0. So again, this would not necessarily be what happens in many cases with protein formulations, but this is an example of a stable protein. Now, with the heat stressing, we were able to see some differences in the particle counts. So if we look at the T0 data, which is in red, we can see that the particle counts in the beginning were actually relatively low. We didn't really see any more than 5 particles in any given vial, and for most cases, there were less than 2. Upon freeze-thaw, there basically were maybe a few more particles on average, but really not a visible particle increase. Agitation was very similar. However, upon the heat stressing, we do see very significant particle counts, especially in citrate forming or citrate containing formulation systems, and also the histidine formulation system. So these are results from the automated system. And then perhaps most surprisingly is that the turbidity actually increased across all of the formulations significantly compared to T0. So if you look at T0, basically we're looking at an NTU of less than 10 or so. There maybe are some slight increases with the freeze-thaw and the agitation, whereas with the heat stress there's a considerable increase in turbidity across all formulations. Now I'll use this time to mention that there was a preservative, an antimicrobial agent of sorbic acid, which is actually FDA approved for such usages, and should keep the bacterial or any kind of microbial growth down, which could induce turbidity. And in addition to that, when I was looking at the chromatography profiles, in the heat stressing, we're seeing the same total area counts between T0 and heat stressing. So this would also suggest that we're not really seeing bacterial or microbial influence on this system, but this is actually really occurring in these vials. And then another interesting result is when we look at our 48 formulations, we see that there's a marked increase in color. So in the beginning, basically both, so at T0, both USP, which is a manual, and automated methods, we're seeing basically colorless solutions. We also see colorless solutions in the freeze-thaw and agitation stresses. However, after the 3 weeks at accelerated temperatures, we see that the increase in color is very significant in all of the histidine samples and also in a few other formulations. Okay, so now we'll go into the conclusion section. And basically, the big take-home message from Freeslate and the automated tool side of this is that this study takes only about 28 hours of attended labor. So, and that includes the formulation of all 192 vials, and it includes all time points. Now, that being said, that's just the attended part. If we look at the formulation, for instance, we can see that it takes about 6 to 9 hours to create these 192 vials, and we formulated in these 2 milliliter serum vials. And of that, it takes about 2 to 3 hours to perform the manual labor that's involved, so setting up the consumables, etc. Now, if we look at the analytical portion of the study, the total time is about, if you do all of the analytics, is about 8 to 10 minutes per formulation, which basically takes about 6 to 8 hours to perform the analysis on all of the formulations, all 48. Now, this doesn't necessarily include the UV or the DLS or the HPLC acquisition times because those can be significant, and it depends on your methods, and it depends on whether you run those in parallel or not. And then another point I want to make is, so in the cases where I was looking at the stress pools, the stress condition pools, I wasn't performing pH or the A280 because sorbic acid was interfering with the A280, and the pH we didn't really expect to change. But what we saw is that still takes about 5 to 7 minutes per formulation, or about 4 to 6 hours for all 48 formulations. And what that's kind of showing is there's an economy of scale here. If you're gonna run, 6, you may as well run 9 different analytics because it really almost takes about the same amount of time, and especially considering that most of it is unattended anyways. Okay, so then the conclusions from the actual performance of the different formulations. So one overarching theme here is that because BSA is so stable, and because these formulations were actually performing relatively well at the initial time point and with freeze-thaw and agitation stresses, really only the heating stressing could give us any information to evaluate the formulation's performances. So the first thing is that the chromatography data, as I mentioned before, suggests that really the freeze-thaw and the agitation stresses don't significantly affect the formulation performances, and that's specifically in regards to the protein's stability. We just don't see very much degradation either from proteolysis or other degradation pathways that could lower the purity, nor do we see significant degradation in the realm of SEC, so we don't see aggregates, for instance, forming. As I mentioned before, the total areas are similar between conditions, which suggests that there isn't anything funny going on with the chromatography or something else happening with the protein that we just can't see. The results from the DLS does provide a little bit of evidence for formulation of small particles in formulations that have lower pH values. Now, in this case, freeze-thaw was able to give us a little bit of information about formulation of particles, but really there wasn't a lot to speak about here, either with freeze-thaw or agitation. Although there was a small correlation between acetate and citrate buffers having real particles and aggregates. And then as far as visible particles, those could actually be pretty significant, and it was basically the acetate and the phosphate formulations that appeared to have the fewest visible particles after the heating process. We also had, we showed that the turbidity actually increases significantly across all formulations, and here acetate actually had one of the lowest overall increases in turbidity. As I mentioned before, the color results were significant, especially in the histidine formulations and a few other formulations. And really, together, what this data is suggesting is that a lot of the stability of these formulations may have actually been due to the non-protein parts of the formulation. And so overall then, as far as looking at the formulation development of stable proteins, which a lot of biopharmaceuticals are, we can see that the automation enables the formulation developers to do very similar studies like this. And as you can see, there is certainly some efficiency gained from these types of studies. We can see that the HPLC, the turbidity, the particle counts suggest that many of the stability factors and stability indicators were probably due to the formulations themselves and not the protein. And I'll take a moment here to mention that all of the materials that were used to formulate were USP or multicompendial grade excipients or buffers. So these were basically the same grade chemicals that would be used in protein formulation laboratories. And one thing that's kind of interesting in the end here is, that these results suggest that with formulation development of highly stable proteins, that looking at the placebos in parallel, certainly at certain time points, may be interesting to see where some of these, where the turbidity, for instance, in particles may be coming from. And that's obviously a mild suggestion because this is one of our first studies of the highly stable proteins using this platform. But one thing that is for sure is that with the placebos, and I did actually create placebos in addition to the formulations, is that the automation allows you to basically perform the analyses of the placebos and the formulations in parallel if one wishes. Okay, and with that, I'll leave it up to a question and answer session. I can see there's already probably a lot of questions. And thank you for your kind attention.
Thank you, Russ. We will now have a brief question and answer session. So the first question, Russ, is could the measured turbidity change be due to a color change of one of the excipients?
At least in part, yes. But one thing that we can see when we look at the turbidity versus the color is that we do see that turbidity is markedly increased in all formulations, whereas the color does change in all formulations, but it increases more drastically with the histidine, and we don't necessarily see that much higher of a turbidity in the histidine than we do the other formulations. So the answer is yes and no. They are coupled, but there's something else going on with the turbidity as well besides just color.
Great. Hey, so Russ, can you tell whether we've ever investigated integration of subvisible particle counters?
I know that we have used HIAC, for instance, as at the very least data loaders. I think that they even are virtually integrated, but it is something that we are still looking into, and I don't know if we currently support the HIAC systems right now.
So Russ, another question is, and I believe you said it during the presentation, but it was, how long does it take to perform such a study?
Right. So then as far as the amount of time, as I mentioned, it really only takes about 28 hours of attended time. Now that's obviously just telling part of the story, right? Because I wasn't including the HPLC analyses. Now, depending on your the methods that you're using and the number of replicates, that can be a significant increase in amount of time needed to perform such studies. Obviously, you also have to wait a few weeks for if you want to do heat stressing. But basically, even with just one or two analysts with an automated system like this, the study could be about as short as a month and let's say 5 weeks, but it could be as much as 2 months, depending on those factors I just mentioned.
So do you know what the accuracy of the 6-tip pipetter is when dispensing 5 microliters and 1,000 microliters?
The accuracy for the 6-tip, I don't think we actually, it's not within our specifications to normally dispense 5 microliters. I think the smallest that we would normally dispense is between 10 and 20 microliters. The accuracy can be calibrated, and the reproducibility from the data I've seen and the data I've seen from our testing and validation is very high. Even at those low dispenses, it's typically within 5%.
So can you tell us how long it took you to design the study?
Designing the study didn't take that long because obviously the intent here was to pull as many different facets from the formulation field and then take as much information from what we can provide to that, so what our automation systems can do. And so it was just sort of the nexus of what are people doing in formulation labs and what can the system do? And that's sort of what decided this formulation study. And I think that really it takes about a day to do that, including making all the software changes necessary to run the systems. But I would say that most people in the formulation field, you can basically use the methods or the previous studies that you've used. You can basically just translate that into the Library Studio language and perform that. Or you could look at our automation and say, well, this really could enable me to do something very, very different, in which case that study planning could take you a day or two to design and possibly do some dry runs.
So this next question uses a term that I'm not familiar with, but I'll read it anyway, okay? And because maybe you're familiar with it. So the question is, can you configure the system to pipette WFI to reconstitute lyophilized products in vials?
Yes, we can.
And then a follow-up question for that is, washing the pH probe and viscometer done with WFI only, and how do you know you've adequately cleaned when using viscous products?
Right, so that's a good question. So we don't use water for injection for cleaning. We actually use HPLC grade water to clean the system. To clean the pH probes. So they go through 3 stages of washing, and then there's an air blade that blows the material off of them. Now, what we can recommend for people who have more viscous solutions is that you can just use control standards between, if you'd like. So you can perform a measurement, and then you can go and clean your pipette tips with the water and the air. You can even increase the number of cycles if you think it's necessary. And then you can measure your standards in between your samples to verify that you're getting the numbers that you expect. And it can also be used as possibly another washing step, if you will. So that's one way to sort of counteract the highly viscous solutions. But if you look at how we wash and dry the pH probes, it's definitely going to take care of all but the most viscous of solutions as is.
So the last question, Russ, is can you explain the advantages of the LIA software?
Sure. So one of the major advantages with the LIA software is that, as I mentioned before, we can actually use the LIA software to run all of these analytics that I talked about. So instead of having to learn the SpectraMax, SoftMax software, and instead of having to necessarily learn everything about the Dynamics software that runs the Diana Pro, you can just use the Automation Studio software to run those systems. You do not necessarily need to know anything about those software programs. Now, naturally, you need to set up the proper methods, but if the methods have already been set up, then you just bring them into Automation Studio. And the same is true with the HPLC system. You don't necessarily need to know how to use ChemStation or ChemLaunch. You just need to have a method set up and Automation Studio, using very similar tasks and coding, if you will, which is really just buttons, can run the HPLC. And that basically is true throughout all of the analytics. So you're basically looking at a very similar design and a very similar implementation for every single analytical method you're using. Now, in addition to that, once that data is collected, you can the databasing is very, very useful because it allows for the ability to keep very good track of what's happened to every single one of your samples, and it's all databased and it's all readily and quickly accessible, which means for notebooking and for presentations, you can decide how much you want to actually, let's say if you had a paper notebook, how much you want to put into your paper notebook, because you may just need to reference the experiment designs and things like that. And if you already have electronic notebooks, then typically we have a way of integrating with companies' electronic notebook systems. So those are two major advantages.
Thanks, Russ. Well, that's all the questions we had for today. If you're still submitting a question, we'll keep the webinar site open for 5 more minutes to allow you time to send it. Unanswered questions will be answered privately in a follow-up email message by Russ. Oh, we just have one more question, so let me just do that. So, do you have airflow visualization studies demonstrating unidirectional airflow within the enclosure?
Not that I know of. I do know that we are trying to put a little bit more emphasis on the aseptic nature of the HEPA enclosure. With the HEPA filters on, there is a powerful fan blowing from the top, and that should be, we can validate that. It's just I don't know what the status of that validation is yet, but I would think that we're already at an aseptic situation inside the hood as far as the airflow from the HEPA filter. Great.
So before concluding today's webinar, I'd like to invite you all to the 2014 Freeslate Forum to learn about the latest in high-throughput research solutions. The Freeslate Forum will feature user presentations that highlight the application of high-throughput research and drug development, workshops and demonstrations of Freeslate's high-throughput research instrumentation, and provide an opportunity to attendees to network with other scientists in their field. The 2014 Freeslate Forum will be on April 8th to the 10th, 2014, at the Hyatt Fisherman's Wharf, 555 North Point Street in San Francisco. And for more information about the Freeslate Forum, contact Carol Gardner at carol.gardner@freeslate.com or visit our website, www.freeslate.com. Also, I'd like to invite you to visit www.freeslate.com to visit our previously recorded webinars on the application of Freeslate's high-throughput research solutions. To see our previously recorded webinars, select Technical Resources from the upper navigation menu, then select Learning Center from the drop-down menu. This concludes today's webinar. On behalf of Russ and myself, I thank you for attending, and we look forward to seeing you at future events. Have a pleasant day.